So you know. Today we have Dr. Daniel Rueckert, who is the Alexander von Humboldt Professor for AI in Medicine and Healthcare at the Technical University of Munich and Professor of Visual Information Processing in the Department of Computing at Imperial College London. He is obviously extremely accomplished in the field of medical image computing. And in particular, he's a fellow of many societies and academies, including the Medical Image Computing and Computer Assisted Intervention Society, IEEE, the American Institute for Medical and Biological Engineering, the Royal Academy of Engineering, and the Academy of Medical Sciences in the UK, and finally, Leopoldina, the German National Academy of Sciences. And today he will talk to us about AI for cardiac image acquisition, analysis, and interpretation, and I will turn it over to him.
Thank you very much, Danielle. Thanks very much for the invitation to be virtually with you today. Let me see whether I can share my screen. Hopefully you can see my slides now. Is that, yeah, perfect, great. So what I wanted to talk to you today about is AI for cardiac imaging, all the way from image acquisition to analysis and interpretation. And I'll just kick right off with some general slides before diving a bit more into the cardiac imaging arena. I guess this seminar series is exactly about this topic here. And I think a lot of the headlines you see look pretty, sometimes perhaps scary. Also, they promise a lot of things. And some of your colleagues at MIT have also written a review, I think last year or the year before, that actually not everything really which AI has promised in medicine has actually worked out well. And in particularly, perhaps the pandemic was one of the examples where AI hasn't really delivered all it has promised. But I'll promise you in the cardiac arena, it's completely different. But in imaging in general, I think it's perhaps at the forefront of AI and machine learning. If you look at this paper, which is now a bit older, but it also has a database associated with it, which tracks all the applications which are FDA approved in terms of being classified as medical devices. It's very clear that imaging is really the vast majority of these applications. So imaging meaning radiology or pathology. So most of the AI tools which are out there really for healthcare are really imaging tools. And in particular, AI and machine learning also has now got more and more attracted interest in the cardiovascular domain, ranging from trying to use AI to identify vulnerable plaque in coronary arteries to general applications in MR imaging. And I think this shows you that there's a lot of potential for AI across the entire imaging domain, but also in particular in the cardiovascular domain. And what I wanted to do today is go through a number of different applications which we're working on in our lab, both in Munich and also at Imperial College in London, ranging from image acquisition. So how can we use AI to enable us to acquire images better and faster, all the way to sort of improving image quality with AI, segmentation, of course, using AI, but also screening and monitoring, as well as disease prediction using AI in the cardiovascular domain. And I'll show you a few examples of all of these different categories. And I just want to start with AI-enabled imaging in the cardiovascular domain. And in order to do that, I want you to take a look at this particular example here. I don't know how many of you are familiar with MR imaging, but the way MR imaging is acquired is you sample a domain called a so-called K-space domain, and then you reconstruct the image by applying a Fourier transform to the image. And what you see here at the top is a fully sampled K-space. That means I've acquired all the frequency information I need to reconstruct the image. And at the bottom, you see a case space which has been randomly subsampled, and only a quarter of the information has been sampled here. And if you apply a Fourier transform to this for reconstruction, you see that the resulting image looks much poorer. Now, why would you sample only a quarter of the information when you really should sample all of the information? Well, sampling a quarter of the information is 4 times faster in terms of image acquisition speed, and therefore you gain a lot of speed in the image acquisition. And in the cardiovascular domain, that's quite important because you typically have to hold your breath for the image acquisition. So making the image acquisition as short as possible is patient-friendly, especially if the patients are quite sick. So a question from an AI point of view could be, for example, well, can we actually reconstruct this fully sampled image from the undersampled image, for example, by denoising the image in the image domain? And I'll show you some older work from our group where we have done that before I also show you a more recent approach, which is perhaps even more powerful. So here the question really is effectively a denoising problem in the image domain. And in order to denoise, we can try to effectively, in the naive setting, you could just say, well, I'm going to use a UNet type structure, whatever your favorite network architecture would be in a deep learning setting, to actually remove aliasing artifacts from the image. And in fact, that works reasonably well, but it isn't perfect. And it isn't perfect because you have no guarantee that the image you reconstruct is actually consistent with the case-based data you have acquired. So you generate a nice looking image, but it might not be a faithful representation of the patient. And that is something which I think in medical imaging is sort of something you cannot really live with. So one approach which we developed a couple of years ago, this was from a PhD student of ours who now works at Hyperfine, which is a company building sort of very low field MR scanners, was the following idea. We basically operate both in the image domain and in the case-based domain. In the image domain, we have a network which denoises the image using convolutional layers. And then we actually move the calculation using a Fourier transform into the case-based domain. You see here in the bottom green box, this F denotes a Fourier transform. Then we perform a data consistency operation. That means we're making sure that the image we have just reconstructed is actually consistent with the case space we have acquired. And then we go back to the image domain. And you can build these very deep networks using intermediate layers of image domain and case-based domain operations. And you can backpropagate through this entire network because this can be trained in an end-to-end fashion. Now, how does this look like when you apply this to some images? So here is a cardiac MR scan undersampled by a factor of 6. Here is a fully sampled MR scan as a comparison. And if you apply this deep learning based reconstruction, you can see you can reconstruct this image with very high accuracy. And actually, turns out that even if you use more aggressive undersampling, here's an undersampling factor of 11, where there's really hardly anything recognizable in the image. The deep learning reconstruction still produces very nice reconstructions. And the reason for this is that actually there's a lot of redundancy in the data because we're imaging the same object over and over again because we want to capture the dynamic information. So there is a lot of redundancy in the data, and therefore the denoising can be done very effectively. So this is something which we've sort of developed and further developed with more refined methods over the last couple of years. But one of our PhD students more recently also thought about whether this problem can be solved in a completely different fashion. And this is what I want to show you here by going back to this figure from the beginning again. And what I've shown you before is that we can actually perform this operation in the image domain. And you've seen that works quite effectively. But actually, the Fourier transform is a linear operation. So I can also, instead of operating in the image domain, I can also operate in the K-space domain and try to estimate the missing lines of K-space almost via an interpolation setting. So instead of denoising in the image domain, what we have done before, we now thought about, can we do this K-space interpolation and operate in the K-space domain? And this has a number of advantages, which I will show you in a moment, and actually probably is practically more useful than the first option. So the reason why this is more useful is because I've shown you before that I've effectively acquired lines in K-space. And this is what we call Cartesian sampling of our K-space line. So here you see on the left-hand side, Cartesian sampling of or undersampling of the K-space. But actually, it might be more beneficial to sample your K-space not in a Cartesian fashion, for example, but in a non-Cartesian fashion. And let me explain you why that might be useful. Well, the most important information in K-space is in the center of K-space. That's where the low frequencies live. And you see here in this radial sampling, which I've shown here, you naturally oversample the center of K-space. And you naturally have a good sampling there, and you have less sampling in the outer regions of K-space. And that is actually a quite good behavior. But if you have data like that, you can no longer apply a Fourier or a fast Fourier transform to your data, but now you have to regrid it onto a regular grid in order to apply a fast Fourier transform, or you have to do non-uniform Fourier transforms, which are extremely slow. So there's a downside to this. But I want to also tell you a bit about how this data is really acquired, because what happens is you have multiple heartbeats. An MR scanner typically operates in a triggered fashion. It's triggered on this R wave. So here you see at the top the EKG or ECG of the heart with the R wave, where the acquisition always starts. Then you acquire some lines of K-space, and then you wait for the next R wave, and you keep on doing this for the next heartbeat, for the third heartbeat, and so on. Okay. And now what you're actually doing is you're pretending that this all comes from the same heartbeat because you have the temporal information, you can align the data, and now you're trying to typically reconstruct from this type of data. And sort of now what you see here is I've basically sampled the K-space domain sparsely in Kx and Y, but also in the temporal direction. So how you typically acquire images here is typically you bin this information into discrete bins. And then for each bin, you have one reconstruction, so one temporal image of the heart. And so the number of bins you effectively have is a temporal resolution of what you reconstruct. And if you make the temporal resolution too low, i.e. you make the bins very wide, then you have motion blurring because the cardiac or the heart is not always in the same phase. So this has all sorts of problems. You lose temporal resolution. You don't really exploit spatial coherence. And the approaches which I've previously shown you, they all require a training data. Here, what I want to show you is an approach which doesn't require any training data at all. It's self-supervised and it's binning free, so it doesn't require any binning, and it's trained only on the data from the subject you're acquiring. And in order to do this, we use something called a K-space interpolation network, which some of you might have come across in form of neural implicit functions. But the basic idea is that I'm taking a multi-layer perceptron, so not a convolutional network, just a simple multi-layer perceptron. and I plug in 3 coordinates, time, kx, ky, and also the coil number. So typically MRs come acquired from multiple coils. You can ignore this here, but effectively you have 4 coordinates. You plug into this network, and then the network aims to predict the k-space value at this point, at this position in time, and at this position in k-space. And then we compare that to the actual sampled k-space value. And then we're basically optimizing this network to predict the sampled k-space values. So this is fitted on the data only from the subject from which we have acquired the data. So after we have trained on the data from the subject, we can now go back to our inference and actually now try to predict the k-space or interpolate the k-space value on a Cartesian grid. Using the same MLP, we now plug in arbitrary values on a grid in terms of k-space, and the MLP will predict the k-space value, and that k-space value can then be used for a Fourier transform and reconstruct the image. So here you see the result of what comes out of this. So this is the proposed sort of reconstruction from 4 heartbeats. So 4 heartbeats is relatively quick. Patient doesn't have to hold their breath for very long. And you can see the reconstruction looks pretty good. You also see some other state-of-the-art reconstruction methods, which I won't explain too much in detail, but you can see that the proposed method works pretty well. And here you see a movie of the reconstruction, as well as some of the state-of-the-art methods for this. Turns out, again, you can push this more aggressively to reconstruct data from only 1 heartbeat, in which case the existing methods typically fail to reconstruct something useful. And you can also see this aliasing pattern now looks very different to what you had before, because we have this radial sampling. You see these rotating lines in the reconstructed image. The proposed method still does a reasonable job, even though, of course, also here now the image quality starts to degrade and is no longer as good as we had in the fully sampled images. But this is a quite nice method because it doesn't really require any external training data, and therefore is also much more self-consistent. So I now want to move on to another topic, which is super resolution, because we have another problem in cardiac MRI is typically that we acquire data slice by slice. I've already shown you that every slice is a breath hold, and you have a limited number of time points you can really here acquire. And typically what happens is you acquire data relatively thick slice, so that in plane the data looks quite good, but if you reconstruct it in 3D, it looks not very good. So here the question is sort of, can you improve the image resolution using deep learning approaches? And you can actually do that. This is some older work where we effectively acquire images in different orientations, in the short axis direction, in the long axis direction, and then plug that into a neural network or in a convolutional neural network, which aims to upsample the images to isotropic resolution. And this works pretty well. And I'm really only showing you this because I also wanted to show you a non-cardiac application of this. So here you see, of course, a cardiac application of this, and you see the deep neural network does a quite good job. You can also acquire 3D data, which is also possible. But if you think about why we don't always acquire the 3D data, I think the last column, the 3D acquisition here, you see the contrast is not brilliant and the breath hold is roughly 30 seconds long. So perhaps most of you can do 30-second breath hold, but I think a patient with cardiovascular disease will not be able to hold their breath for that long, or they don't have a disease. So, so I just wanted to show you that in principle, you can use very similar approaches also for fetal imaging. This is some work which we've been doing with colleagues in London, where you acquire, similar to the cardiac data, you acquire images of the fetus in real time, And the problem is that the fetus obviously moves during the image acquisition. So in addition to just doing super resolution, you also have to do motion compensation and estimate how every slice is related to every other slice. But the deep neural network can also do that. And the way we do this is by acquiring lots of data in one orientation, as you see here. And then, for example, a second stack in a different orientation. And we keep on doing this for a couple of times. And then if you take that data, you can effectively use a deep learning approach also to reconstruct this fetal data and correct for every slice or estimate for every slice where in the 3D anatomy that slice really belongs to. And of course, that only works if you have a rigid object, which in the case of the brain, that assumption is reasonably valid. So let me move on to AI-based segmentation. And I guess if you use a UNet for segmenting the heart, you're not doing something very groundbreaking, because probably everybody else in the world is also doing exactly the same thing. But I also wanted to show that actually, if you have a lot of training data, you can actually, these quite simple approaches in terms of UNet can actually work really well and produce very reliable segmentations. I wanted to also show you something which we've just presented at MICCAI a couple of weeks ago, which tries to tackle the problem that our data set is typically acquired as a set of sparse long axis and short axis planes, but we really would like to have a 3D or 4D segmentation, and not only defined in the imaging plane. So what you saw here is a segmentation just defined in these planes, but I don't really have a 3D model from this. So I want to come back to this idea which I showed you for image reconstruction, where we've used neural implicit functions. And I want to show you an approach where we use neural implicit functions for segmentation. And I think you're all familiar that typically we have our signals, when we acquire them, are sort of discrete in terms of pixels or point clouds. We can also try to come up with an implicit representation, for example, of a segmentation. And here, the idea is quite simple. I try to represent an object using a sign distance function. So if you see this bunny here, everywhere inside the bunny, the sign distance function is negative. Everywhere outside in the background, it's positive. And where the sign distance function changes from positive to negative, I have the boundary of the object. And now I can use a MLP in the same way I've used it before in order to try to represent this object. So here, the neural network is not convolutional. It just takes X, Y, Z as input and gives me the sign distance function value as output. And that's called a neural implicit function. And that function obviously can represent shapes with an arbitrary precision, because I'm no longer living on a grid. I can put any X, Y, Z coordinate in there. So how do I use this for segmentation? Well, first of all, it has a couple of approaches why this is good. First of all, if I have a sparse set of planes which are not parallel to each other, but for example, in an arbitrary orientation, it's very hard to, for example, define a convolution which basically takes information from a long axis and short axis slice into account at the same time, because basically my data is no longer on a Cartesian grid. And I can also not easily do, for example, extrapolation between imaging slices or beyond imaging slices. So the idea which we've used here is that we're using, this is actually related to an idea from Brano, I think is also in the audience here, when he did his PhD with Björn Menze in Zurich, that we try to basically formulate a problem similar to a shape reconstruction problem, where we're trying to find a latent code which reconstructs a shape or a segmentation, and that reconstruction is fitted to an image. So let me show you this perhaps more simple in this diagram here. So we have a MLP which has two inputs. It takes in XYZ coordinates, but also a latent shape representation, which is learnable. And then we can fit this to, in the training phase, to both images, which we have, segmentations, as well as intensity images. So the segmentation branch is effectively fitted to segmentations, and the reconstruction branch is fitted to the image intensities. And of course, we can only sample XYZ coordinates which are either on a short axis or a long axis slice. So in the forward pass, we simply plug in these coordinates and the latent code and observe the segmentation label or the reconstruction label. For training this, of course, we have to backpropagate, and we have to backpropagate with respect to the latent representation, but also the XYZ coordinates. Now, during the test time, the MLP is now frozen. We can no longer change the MLP. The MLP weights are given. We can only change the latent representation. And of course, at test time, we also don't have the segmentation. We only observe the image intensity. So what we effectively do is we randomly initialize our latent code, and then we are backpropagating through the frozen MLP to optimize this latent code so it fits the image intensities. And once we have done that, we can now plug in with the same latent code the XYZ coordinates, and we obtain both the segmentation label as well as the intensity information at that point. And here's a few visual examples of that. So you see, for example, that the intensity images which we're reconstructing look a bit blurry, but that's not really our problem, because we don't really care about the intensities. We only really care about the segmentation label. But the segmentation label is now defined for any XYZ point, even if it's not on a sampled grid. So I think that's a quite nice approach, because we can now reconstruct a 3D object with arbitrary resolution. We just have to define the grid on which we want to evaluate the neural implicit function. I was just showing you also an application of the segmentation pipeline to a large data set. We have worked a lot with UK Biobank, because UK Biobank is collecting information from a very large number of subjects. I think this is now 2 years old. I think they have scanned now over 65,000 subjects. And for each individual, they acquire a whole body MRI, brain MRI, including diffusion and functional imaging, cardiovascular imaging, and a lot of lifestyle genetic information, as well as clinical outcome data as it becomes available. But keep in mind that this is a sort of normal population, not a disease population. And now if you have, for example, an automated segmentation pipeline, you can apply that to all of these 40,000 subjects. You can sort of, for example, derive phenotypes compared to different, for example, how these phenotypes vary with sex and age. But also you can do something similar to a GWAS study. You can now, for example, correlate all these imaging-derived phenotypes with respect to other information you have in UK Biobank, for example, lifestyle, alcohol, smoking, physical activity, and so on. And you can effectively investigate the correlation between these different factors. I should really point out that this is really correlation, so you have to be a bit careful how you interpret this. And perhaps sort of towards the end, I also wanted to show you some applications for outcome prediction. Here, we're trying to use the pipeline we have developed before, which produces a segmentation, to predict outcome in patients with right ventricular pulmonary hypertension. So here you see the segmentation obtained in cardiac MR images for different cardiac phases and different short exercises. Since we can also sort of relate these images across time, we can also extract the motion trajectories of these points. And what you see here as these spaghettis coming out of the right ventricle, these are the motion trajectories of the surface points of the right ventricle. And we can now track this motion of the heart, but also use that as a feature for outcome prediction. And the outcome prediction model, which we've initially tested, we've now further developed, but I'll show you just the basic idea is simply to train an autoencoder model, which takes these cardiac shapes and the cardiac motion and compresses this information into a latent code. But we are also forcing this latent code to be predictive of survival of these patients. So we have a Cox regression model here, which predicts survival. And we're forcing this latent code to be meaningful for survival by optimizing both the reconstruction loss and the survival loss. And if you then apply this to cardiac MR data, you see that you get a much better separation between two patient groups, those with good outcome and poor outcome, compared to just using standard parameters from cardiac MR, such as ejection fraction or ventricular volume. So I just want to sort of highlight one or two problems which we're still facing before I sort of conclude. I guess we are all sort of aware that we have a lot of biases in our data set. So if your data set looks like it's coming from this population, it's perhaps not as representative as if it would come from a more diverse population. And there are a number of papers, not only in the machine learning community, but also in the more general science community, which have highlighted that this is a huge problem. And it's not actually a new problem which comes from AI, but it's probably amplified by AI. But this problem has existed before. And if you don't know this paper here, I would strongly recommend take a look at this. But also, we have other problems related to that. For example, if I train my model on UK Biobank, it will probably look a bit like this. And then if I give it to my local cardiologist to deploy, they apply it to data which looks like this. And that typically breaks your models because your models cannot adapt to, for example, that degree of pathology if they have not seen this pathology before. And of course, this is not only pathology, but can also come from different imaging hardware, different imaging sequence, or even different imaging modalities. So this is something which we're still working on tackling on. And I just want to show you, actually, let me just skip this because I think I just want to show you that there are a number of different approaches for addressing this. We started off by trying out adversarial approaches, which, for example, try to make sure that the features you learn in your neural network are invariant, for example, to scanner changes. And this can be done by having an adversarial component in your loss. Then we've tried out different ways of doing data augmentation in different forms. You see here two papers from our group which try to do that. And all of these approaches, if you take them together, they sort of tackle this problem reasonably well, but there's probably still room for improvement. And I think the best solution to the problem is just a very, very large data set. And I think you also see this in some of the more recent work on foundation models, which illustrate that there's probably no cure for having a very, very large database. So I want to come to an end here and just sort of briefly highlight a bit what we're doing next. You also saw this debate a couple of years ago, Jeff Hinton saying we should stop training radiologists now in 2017, and Curtis Langlord saying, no, we should we need radiologists, but AI will not replace them, but radiologists who do AI will replace those who don't. I much more subscribe to the second statement, but perhaps even that hasn't come true in the last couple of years. There's also another interview with Jeff Hinton from, I think, a couple of months ago, where he said, I was right all along. It was just not 5 years, it will be 10 years when we don't need them anymore. Perhaps this is also not quite right. I doubt, at least I'm pretty sure that this prediction will also be fundamentally wrong. And actually, at the moment, we have a huge shortage of radiologists, as you see on the right-hand side. So my take on this is a bit different. So at the moment, we're doing sort of things in a very serial fashion. We acquire images, we reconstruct them, we analyze them and interpret them. And all of this can be done with AI to some degree, but it's a bit suboptimal because everything is done in isolation. I think with AI now we have the opportunity to sort of couple the processes more tightly. For example, in the reconstruction, I could say, if I don't have enough data to reconstruct, then I need to acquire a bit more data, correct? I can steer the scanner. And similarly, from the analysis, I could sort of say the image is not well enough reconstructed for analysis. You need to improve the image reconstruction. So, and then the reconstruction might say, okay, I need to acquire more data. So this close coupling of acquisition, reconstruction, and analysis and interpretation is something which I think is crucial, but also the ability to have interaction between these components and optimize this entire pipeline with respect to what you clinically really want to know. And perhaps that then sort of leads naturally to the question is, do we actually need images at all, or can I just have a very large neural network? Since each of these components could be a neural network, I might just chain all of this together and have a very large neural network solving that all. And I think that is, a very appealing idea, but an idea which only ever will work if we also can guarantee performance of such approaches. So with that, I'll stop here. I just want to acknowledge some of our funders, but also mostly importantly, the colleagues both in London and in Munich who helped doing all the work. And if you have any questions, I'd be very happy to answer any questions you might have.
Great, thank you so much. Let's see if there are any questions from the audience. Feel free to just start talking and we'll see how that goes. Anyone who has a question.
Great talk, Daniel. Thank you so much. I have one question about your vision that you presented at the end. What are your thoughts about the endpoint definition? Because like the part that you link diagnosis to imaging acquisition, like, do we need to have questions baked in? Because it's sort of like, you need to know what you're looking for.
So I think you're absolutely right. So you hit sort of a sore spot. I mean, I think there are two applications where you can use imaging. One is sort of imaging sort of in a radiology department in a hospital, where quite often you don't quite know what you want to measure, correct? You don't know because a patient has some chest pain, and you don't know whether exactly you want to effectively use imaging as an explorative tool. And there, I think you effectively need to optimize for the image quality. So I think it's not directly the clinical endpoint you would optimize for, but you should optimize for image quality. But then there are other applications, for example, in screening or in other scenarios, like in UK Biobank, when you do population imaging. There you typically, for example, in screening, you typically predefine what you want to measure, and you only care about measuring that thing, except for incidental findings. So in a screening setting, you could, for example, say, I would like to measure whether there are, for example, lesions in the breast, or in the cardiovascular domain, I want to measure ejection fraction, right? And then you could sort of optimize your acquisition for that. I also think that in general, it would be quite useful to be able to optimize for a downstream task such as segmentation, correct? You might say, actually, I want to make sure that I acquire an image which is optimally suited for example, for segmentation. Perhaps you can also reconstruct the same data in another way that it's optimally suited for visual inspection, right? So you can almost sort of think of this as a multitask network which tries to optimize multiple tasks at the same time. So that's sort of, but you're right that for a number of scenarios, I don't know in advance what I should optimize for.
Thank you very much.
We have another question in the chat. So the question from Steve Choi was the random sampling in Cartesian coordinates required? And also, did you consider the redundancy of multi-coil dimension or just coil combination for denoising for a fast scan?
So, the question, does it refer, I mean, if it refers, well, for both cases, we did random sampling. In the Cartesian case, just random sampling of lines in K-space. But for every time frame, we sample a different set of lines. So you get very incoherent artifacts. For the radial sampling, the sampling is also random, using something called a golden angle acquisition, where effectively you always offset one radial line by a certain degree to the next radial line. And that sampling pattern has been shown, if you use a certain angular offset called the golden angle, then you can actually guarantee that you get a relatively uniform set of radial lines, no matter how you sort of later on bin it. So that is what we used for this. And we simply effectively measure the consistency, or in this neural implicit function, we model the neural implicit network tries to model all coil images at the same time. So the coil index goes in as one of the input coordinates to your network. If that was a question.
Great, thanks. I had a question also as well about the survival analysis work based on the shape and motion of the heart. So as far as I understood it, you're throwing away all of the intensity information in the scan and just looking at the shape and the motion. Did you investigate whether incorporating intensity information in some way would be helpful, or did you decide that that wouldn't be applicable for you?
Like a radiomics type approach, or
well,
something?
I can imagine, I don't know, for example, if there's any hyperintensities or hypointensities in the heart, if that's indicative of future problems.
So we didn't do it, but in principle you can do that. I think our observation was that the data came from multiple centers. So I think there's quite a lot of variability in terms of image intensities if your data comes from multiple centers. And so we use the segmentation as a way of basically abstracting away from the image directly to get to something which is more sort of robust across different clinical sites. So we did this on purpose, but in principle, I think you could do that quite easily and sort of basically incorporate also intensity features here.
Got it. Okay. Any other questions from the group? Okay. Well, I'm going to give you all, I formatted it all now. The links to join the mailing list. We've been sending out advertisements for this new series to a lot of different places, but at some point we're going to stick to that list. So if you'd like to keep attending, then please join the list. And we also have the speaker suggestion form for anybody who wants to suggest who should come. So I wanted to thank you all again for coming, and especially thank you to Dr. Rueckert for presenting. And we will see you next month. Thank you very much.